My name is Ralf Bartenschlager.
I am a molecular and cell biologist by education. During my studies, I focused on viral infections. I'm head of molecular virology at the Heidelberg University, more precisely at the University Hospital. At the same time, I also head the Department of Virus-Associated Carcinogenesis at the German Cancer Research Center, which is also located here in Heidelberg.
At Ralf Bartenschlager's institute, research is conducted on hepatitis C viruses and on tropical viruses transmitted by mosquitoes, such as dengue or Zika virus.
Also die Tätigkeiten im Labor.
The activities in the laboratory can be defined in different ways. It is, of course, experimental research, in other words, basic research. How are cells infected by these viruses? How do these viruses penetrate the cells? And above all, what changes do these viruses cause in the cells? That is what determines how severely a cell is damaged, and thus how severely an organism is damaged. We are also interested in how these viruses assemble, how they leave the cell, and how they infect new cells. In other words, how they spread in the organism and in organs. Another very important point is how the immune system reacts to the infection.
The breakthrough in hepatitis research was to be able to create a corresponding cell culture in the laboratory. Ralf Bartenschlager and his team succeeded in doing this.
Wenn man Therapien entwickeln möchte,
In order to develop therapies, you need cell systems. Otherwise, you cannot test whether a substance has any antiviral effect at all. We succeeded in replicating parts of the virus in the cell laboratory. That was the starting point for therapy development. In principle, all medications that are given to patients today have been developed with this cell system.
The scientists working here at Ralf Bartenschlager's institute adapted their research fields and goals to current requirements during the corona pandemic.
am Anfang
At the beginning of the pandemic, we focused primarily on things that would benefit clinical practice, detection methods for antibodies and the virus itself, and above all, neutralization tests. We were asking the question, do the antibodies actually protect? At the same time, we also ventured into new research topics.
For his fundamental successes in the field of hepatitis research, Professor Ralf Bartenschlager has been awarded very prestigious scientific prizes, among others the Robert Koch Prize, the Lasker Award, and last but not least, the Hector Science Award.
die Verleihung des Hector-Preises.
The Hector Science Award and the associated membership in the Hector Fellow Academy is also a very high recognition of our work. Besides the prize, it also has the advantage of becoming a member of the Hector Fellow Academy. The Hector Fellows cover a very broad and interdisciplinary field, from particle physics to polar expeditions. Truly a very wide spectrum. This interdisciplinarity is really its strength. One starts, even more than one already did before, to think beyond disciplinary boundaries, to develop projects and to initiate collaborations. This is, of course, a wonderful opportunity to open up new fields of activity.
Another opportunity that the Hector Fellow Academy offers are doctoral positions for young scientists from the STEM subjects, medicine or psychology, just as Yannick Stahl, who approached Ralf Bartenschlager with his own research idea.
Also Yannick Stahl.
Yannick Stahl had applied with the idea of working on the topic of SARS-CoV-2. His idea was to research what changes the virus causes in a cell after infection. That convinced me. But what convinced me even more was Yannick himself, his personality, the qualities he brings with him, and his interest and motivation. These are very important points.
Mein Name ist Yannick Stahl.
My name is Yannick Stahl. I am a doctoral researcher in the research group of Ralf Bartenschlager. I am working on the SARS coronavirus 2, and to be precise, I am trying to study the pathogenesis of the virus and to find new host cell factors that favor the replication of this virus. The Hector Fellow Academy offers a very diverse environment of experts from different disciplines who are more or less close to my own research, but who can also provide very different insights. They are able to ask questions that, if you are in virology, you would never think about otherwise. You get in contact with these people at networking events at the Hector Fellow Academy. That is very useful for me, and of course it opens up whole new insights for my own research that I wouldn't have been able to recognize in the first place. The interesting thing is that the system we are looking at is very complex. It's not just a single cell we are dealing with, but a cell that in this case is infected by a parasite, a virus. For us, identifying the host cell proteins that are necessary for the coronavirus to replicate is just the first step. The goal of our work is to provide new approaches for medication development against COVID-19. And I would like my research to be part of this effort as well.
Wir müssen, wenn die nächste Pandemie kommt,
when the next pandemic happens, we need to be better prepared. This includes the development of broad-spectrum antivirals, substances that are able to fight not just one virus, but a whole group of viruses. For example, a corona medicine or a flavivirus medicine. This means that if the next pandemic were to be triggered by a coronavirus, we would immediately have a treatment ready that could then be given to people who are seriously ill, for example.
und dann beispielsweise schwer erkrankten Leuten dann auch geben könnte.